Well, hello everyone, and welcome back to Science in the Age of COVID-19. Today we have a very special seminar for several reasons. This is the 42nd episode of the seminar series. And as we all know, 42 is the answer to life, the universe, and everything. The second reason that today is so special is that our speaker volunteered. And this is exactly the sort of interactions that we've been hoping to generate with this seminar series. So this is a reminder to all of you watching, if you have an exciting story to tell, or if one of your colleagues has an exciting story to tell them to reach out to us and we will get them in the lineup. So today's speaker is Mattew Parker from the bioinformatics core at the University of Sheffield in the UK. Matt has done a lot of research over the years, mostly on the genetic underpinnings of various nasty cancers. And the Sheffield core is really good at genomic surveillance of various things going on. So when the pandemic started to blow up, they were watching all the samples and were among the first to detect the D614G mutation, and later verified that it indeed was more transmissible, and then followed that up with the more insidious B.1.1.7 variant of concern. Today, Matt will probably tell us a little bit about their surveillance and a lot about a particular phenomenon, which is subgenomic RNAs of the ancestral virus and the variants. Okay, Matt, thank you.
Hi, thanks for that great introduction. Hello, everybody. Yes, so I'm Mattew Parker, and I'm from the Sheffield Bioinformatics core and the Sheffield Biomedical Research Centre. And I'm going to talk a lot today about subgenomic RNA in SARS-CoV-2, and particularly in B.1.1.7 infections. So we'll start off with an introduction to what we've been doing in Sheffield for SARS-CoV-2 sequencing. And then we'll move on to subgenomic RNA and what it is and how we detect it in our data, and then finish on applying that technique to B.1.1.7 SARS-CoV-2 viruses. So just before I start, I know we usually do acknowledgements at the end, but this is such a big team effort across Sheffield and across the wider country in the UK that I thought I'd start with this. So we have a genomic sequencing group in Sheffield, the SARS-CoV-2 genomics group, led by Dushan De Silva and supported by me in the bioinformatics core, and lots of others across the university and across the region supporting our sequencing effort. And we joined COG-UK at the start of the pandemic to really share our sequencing data with the rest of the country and the rest of the world. So they provide funding for this project. So our first genomes were last March. Actually, Dushan emailed us one night in late February if we'd help sequence the first two samples that had arrived in the region. And myself and Matt Wiles are involved in nanopore sequencing in the university, and we jumped at the chance to help sequence these genomes. So on the 11th of March 2020, we received those samples and prepped them. And by 7 p.m. that evening, we'd loaded the Oxford Nanopore GridION with those genomes. And by that next night, on the 12th of March, our genomes were live on NextStrain and GizAid. And you can see them in this tree on the right, where the tree looks very different now, the global tree, to what it did back in March last year, with very few samples, but you see the first two Sheffield samples there. So we're really lucky in Sheffield that we've got a really nice joined up infrastructure between the university and the hospital, which I think really helped our success in being able to sequence samples quickly, because with genetic epidemiology and surveillance, we need to be able to to link our genomes to metadata that makes that sample useful, things like date of collection. And so we have a really nice metadata flow from the diagnostic lab, where they send us daily dumps of all the patients that have tested positive in the hospital. So we get that on a daily basis. That's all automated, and that goes into our LIMS system. And then we also get the positive samples. So these samples are tested by usually by RT-PCR, and the positive samples are then shipped to us, where we extract RNA. We do the Arctic Network PCR protocol, which I'll talk about in a couple of slides. We then do nanopore GridION sequencing, and then the Arctic Bioinformatics standard operating procedure, which I'll also talk about. And then we upload these genomes to COG-UK, and we've uploaded around 8,500 sequences to date, and usually within around 48 hours of receiving that sample in our lab, and 72 hours from that sample testing positive, being collected from an individual. So we can really turn around sequences pretty quickly. And this is just the GridION in the back of my boss's car, because we were really worried about getting locked down back in March and not being able to do any more sequencing. So we transferred the GridION over to Dushan's lab, where we've continued to do the sequencing since. So that's our pipeline, and that's what we've been doing to do our genomic surveillance. And really, we started with those 2 samples and then quickly escalated to over 100 a week throughout most of 2020. We had a little bit of a lull last summer where numbers in the UK were quite low. And then into the autumn and winter, where we had our second lockdown in the UK. And then you see numbers increasing dramatically over the Christmas period, when the B.1.1.7 variant emerged. And really, we needed to respond quickly and sequence lots of genomes to see how this was spreading in our local region, and help nationally track that variant. So we quickly scaled up. And, really, I think a lot of groups around the world are using this Arctic protocol, and we really owe a lot to these researchers for providing this really nice protocol for sequencing the SARS-CoV-2 genome. We're able to order the primers, sequence the genome very quickly, and get the data out there so that people could use it for genomic surveillance. So the way this works is you take a swab sample from a patient and you extract RNA, and then you do RT-PCR to produce cDNA. And then the approach is actually tiled amplicons. So this is the whole SARS-CoV-2 genome on the bottom. And you see each of the Arctic Network amplicons from 1 to 98. So there's 98 amplicons covering the whole genome, and they're split into 2 pools. So there's an odd numbered pool and an even numbered pool. So you do the PCR of those 2 pools separately. You clean them up and combine. And then we run on the GridION. Now we're doing around 47 samples per flow cell plus 1 negative control. And, a lot of people ask why so many amplicons. I think it's, this protocol was designed for very degraded RNA. So initially, I think for Ebola genomics. So this enables us to get full genomes, or at least as much of a genome as we can from that sample, because the amplicons are so small, so around 500 base pairs each. So it gives you the best chance of getting a really nicely high covered, a nice complete genome for these SARS-CoV-2 samples. So once you've sequenced it and you've got your data, the bioinformatics protocol involves filtering the reads based on quality and length. And this filtering step on length is really important, and I'll talk about this in a couple of slides. So it's important to remember that the normal protocol does this length filtering. Then do primer and adapter trimming. This is really important to remove issues that, there's a few variants that were called initially in the pandemic by certain groups that had not trimmed primers, and those were then put into public databases and actually found to be false positives. So it's really important that you follow this protocol so that we don't get any of those in the public databases. You map these to a reference genome. You call variants based on that reference. So the reference is the Wuhan reference. You determine the changes in that reference. You filter based on quality, and then you can generate a consensus sequence based on those variants. And it's really nice. The Arctic Network people have put together a really nice bioinformatics package for this, and it's a couple of commands to do this to get from your sequencing data to a final consensus sequence. So really, we actually got involved in SARS-CoV-2 expression, I think probably late spring last year, early summer, when we were approached by Silvana Guderera and Simon Malal of Western Australia, and they were interested in looking at SARS-CoV-2 expression from genomic sequencing data. And they approached us, and we started thinking about developing a tool with them to try and detect this data. Expression in our genomic data. So So as a bit of a background before we go into how we do this, I thought I'd tell you about how subgenomic RNAs are produced and how SARS-CoV-2 expresses its genome. So this is a figure of SARS-CoV. It's not CoV-2, but this is a SARS-CoV-2, but the method is the same. So SARS-CoV-2 has a positive stranded genome. So this is the genome here, 5' to 3', so from left to right. And it's organized into these open reading frames. So ORFs 1A, 1B, spike protein, 3A, 3B, envelope, membrane, nucleocapsid, etc. And this enables expression of all the proteins that SARS-CoV-2 needs to evade the immune response, to replicate, to produce new virons, etc. What's an important feature that I want you to know is this leader sequence. So this leader sequence is present at the 5' end of the genome. And this leader sequence contains what's called a transcription regulatory sequence, or TRS. And this TRS-L, so it's in the leader sequence, is really important for this discontinuous transcription that SARS-CoV-2 carries out to express its genome. So there's this TRS-L in the leader sequence. Now, throughout the body of the SARS-CoV-2 genome, at some of the ORF start sites, so for example for spike, there's another TRS, which is identical to the TRS in the leader. And this TRS is called TRSB, so it's a transcription regulatory sequence in the body of the genome. Okay, so you've got these two TRS sequences. And what happens is RNA-dependent RNA polymerase, which is encoded for by SARS-CoV-2, starts transcribing a nascent negative stranded RNA from the 3' end of the genome, and it travels along until it hits one of these TRS sites. Then what it does when it hits this TRS site is it switches template from the body of the genome to the leader. So you get this jump, this discontinuous transcription, whereby you get the leader sequence incorporated into this new subgenomic RNA molecule. So there are several of these, and the subgenomic RNA for S includes all ORFs downstream, and as you move more 3', the subgenomic RNAs become smaller. So it's really important to note that all subgenomic RNAs that are produced in this way have a leader sequence at their 3' end, their 5' end, sorry. So there are actually two steps to this. So the initial production of a negative stranded pre-subgenomic RNA by RDRP, and then you get positive strand synthesis to produce your final subgenomic RNA. And we need to know that for some of the final slides in my presentation where we talk about a mechanism for recombination. So we'll talk about that later. So you get these subgenomic RNAs produced, and it's thought that this is because these two TRS sites are brought together by an unknown protein complex. So at each of these TRS sites, they're brought together in close proximity. So when RDRP is producing this nascent negative stranded RNA, it hits this TRS site and can jump to the leader sequence in the genome and transcribe the rest of this molecule from that 5' end. So all subgenomic RNAs contain this leader sequence at their 5' end. And we wanted to know if we could detect this in our reads. So the interesting thing is, I want to say by chance, but it's not really by chance, because the Arctic Network protocol is designed to tile the whole genome, right? And it just so happens that primer 1 starts at the start of the genome, but it is the sequence, primer 1 is the sequence of the leader. So actually what happens in the Arctic Network PCR protocol, as well as amplifying the genome, which is the intended product of this reaction, you actually also produce unintended amplification of subgenomic RNA, because primer 1 encodes the leader sequence, and all subgenomic RNAs have the leader sequence at their 5' end. So for example, if we look at this plot here, we'll look at the N subgenomic RNA for SARS-CoV-2. I've put a line here where this hits the genome, where this TRS site for N hits the genome, and you can see it hits the genome in amplicon 93. So what happens in N's case, N subgenomic RNA, is you get amplification from the leader sequence of the N subgenomic RNA from primer 1 in that pool to the end of amplicon 93 to the normal primer that you would have at the end of that amplicon. So you get this unintended PCR product, and this happens for all of the subgenomic RNAs that are the canonical subgenomic RNAs, so those produced from known TRS sites in the SARS-CoV-2 genome. So it's actually a really nice byproduct of just getting a consensus sequence. This data comes free with the Arctic Network Protocol. So this is what the raw data looks like, and initially it's quite complicated to look at. So we're going to start on the right-hand side here. So this is data for amplicon 91 at this right-hand side. And each one of these bars here is a sequencing read. And the bit that's all blue here, this matches the genome. So this is a normal amplicon from the Arctic protocol, which is unfiltered and untrimmed. So this region at the end of the read here is called a soft-clipped region. This is what the mapping algorithm does when it can't match the reference. And this actually here is adapter, because we've not trimmed adapter in this data. So that's normal. It covers the whole of the amplicon. If you imagine the amplicon goes somewhere down here, these reads cover the whole amplicon like you would expect from this data. Then what happens for subgenomic RNA? In this case, I'm looking at the ORF6 TRS site, which hits amplicon 89. It hits actually in the middle of amplicon 89. And then you see these reads here that have bits that match the genome. So this part, this part here. And at the right-hand side, this is adapter sequence. And then you see here this soft clipping that occurs in the middle of this amplicon. And normally that wouldn't happen from the genomic reads. They would carry on for the full amplicon. And this soft clipped area, the part of the read here that doesn't match the reference, this is actually a leader sequence. So this matches 100% to leader. And then there are other reads that support subgenomic RNA that actually come from the adjacent amplicon, and we think this is maybe unidirectional amplification. But either way, you end up with reads that have a leader sequence attached at this end, so at their 5' end. So the one thing to note here is obviously that these amplicons are smaller than you would expect from the genomic, and they also have this bit that doesn't match the genome. So the normal rules of next generation sequencing don't really apply. So we don't want to trim adapters or primers in subgenomic RNA analysis, at least of this data, because we think that they can also that process can remove the leader and stop us identifying the subgenomic RNA reads. So if you take those reads that we've classified with our tool, and I'll talk about that in a second, into genomic RNA and subgenomic RNA, you see the genomic RNA is really nicely peaking around 500 base pairs. So this is just plot of read length. So you see 500 base pairs for all of these amplicons. And you see things like amplicon 89, which we just talked about for ORF6. You see ORF6 subgenomic RNA is much smaller. It's actually around 350 base pairs. And the Arctic Network SOP, the bioinformatics SOP, is designed to produce the best consensus sequence and not retain subgenomic RNA, because it's not what it was designed to do. So there, you actually, if you follow that SOP, you would filter out these reads because they're less than 400 and sometimes larger than 700. So we need to do no length filtering of our data to be able to call these subgenomic RNAs. And so we developed a tool to detect this, which is now published in Genome Research. It's called Periscope. It's a really bad pun on subgenomic, submarine, and we're kind of searching for things, so we called it Periscope. And this does more than just classify. So it does this nice thing where it tells you which of your reads are subgenomic RNA or not, and it enables you then to go and look at the data and see which of your reads are subgenomic RNA. But we also do some normalization. And so it's important that we normalize the levels of subgenomic RNA that we're seeing to the amount of sequencing data we're producing. So you can do it several ways. And we found the best way actually was to normalize our data to the number of reads resulting from that amplicon, because we want to kind of try and control for amplification efficiency some way, because obviously some amplicons produce better data than others in these protocols. And so we wanted to make sure that our subgenomic RNA was the best representation of that abundance that we could from the data that we have. So this tool's freely available. You can go ahead and download it and run it on your data, and you can, let me know if you've got any problems. We're happy to run your data through it too, if you would like us to collaborate. So please get in touch and visit the GitHub and download it and try it out. So we did this, and I think, just to show that we can detect most of the SARS-CoV-2 open reading frames with this technique, We can see most of them apart from ORF10. And actually the literature where people have done specific experiments to look at expressed RNA, like direct RNA-seq, they see very little of this open reading frame too. And we see 1 or 2 reads in a few of the samples. So this is about 800 samples here. And there are a few that I've actually manually reviewed, and they do look like they do have 1 or 2 reads supporting ORF10, but it's a very, very low level compared to the rest of the open reading frames. And, you know, like the literature, we see large amounts of subgenomic RNA for M, N, S, ORF6 and E. We see less ORF7 than we would expect, and we think this might be due to amplification problems, because remember that, you know, the RT network protocol is not really designed to look at subgenomic RNA, but we can definitely use this data to get added value and look at subgenomic RNA expression profiles. So again, this is normalized data, so we normalized to genomic reads from the same amplicon. So that's interesting, but now we can start to do comparisons, and we looked at another data set from Glasgow just to show that it wasn't a site-specific phenomenon, that we could detect this subgenomic RNA, and we see very, very similar patterns from other data sets. And then we went one step further, and we collaborated with them on a cell culture experiment. So they used this SARS-CoV-2 toolkit that was designed at Glasgow by Suzanne Rinn, and they they infected Vero-E6 cells with 3 different SARS-CoV-2 viruses. They're actually very early in the pandemic, so they're pretty similar compared to today's different variants. And they infected Vero-E6 cells, or Vero-E6 cells expressing ACE2, the receptor for SARS-CoV-2 spike protein, and then ACE2 and TRM-PRSS2, a protein that's involved in infection. So you can see over the time course, the blue line is Oxford Nanopore Arctic data, and then the orange is Illumina metagenomic data. So actually this is unamplified, unselected data. It's just all the RNA in the pot. So actually we get a really nice correlation between Illumina metagenomic and ONT Arctic. And you can see across the time course, we see an increase in subgenomic RNA in wild type. And then if you express ACE2, you start seeing changes to these kinetics, especially in this virus, PHE2 down here. You see maybe a peak starting to occur at 48 hours instead of 72. And then in ACE2 plus TM-PRSS2, we see this definite change in the subgenomic RNA kinetics. We see an increase and a peak at 48 hours, which drops away at 72 hours. So it's nice that we can apply this tool to get real biological answers from this data, you know, and it's obviously very, although it's amplified and it's a byproduct of this sequencing reaction, we see good concordance with Illumina metagenomic sequencing data too. So we really started on this journey. I mentioned Silvana and Simon, and they contacted us about subgenomic RNA early in the pandemic because they'd noticed a non-canonical subgenomic RNA in SARS-CoV-2 viruses that actually had this mutation in the nucleocapsid gene. So this mutation is R203K G204R, and it's a triplet mutation. I'll talk about that in a second. First, I thought I'd explain what non-canonical subgenomic RNA is, and I think the definitions vary, but in our case, we define it as subgenomic RNAs that have leader, because they're the only ones we can detect from our sequencing data. They have leader, but they're not produced from a canonical TRS site. So there's no TRS site present at the point at which RDRP jumps from the body of the genome to the leader sequence. And so I've removed all the canonical subgenomic RNAs from this plot. And you can see in our data set, we've got a lot of these non-canonical subgenomic RNAs spread out across the different open reading frames. You do see a lot of clustering around known open reading frame start sites. And I think that's just because we're defining a canonical subgenomic RNA as plus or minus, I think, 20 base pairs from that TRS site. And so there are some that are slightly outside that range where RDRP maybe just jumps a few base pairs later or a few base pairs earlier. So you get these kind of staggered subgenomic RNAs that probably are canonical, really. And then you do see them throughout the ORF, and you know, in 3A, for example, there's this really nice cluster here, and actually that corresponds to ORF3B. So tantalizing possibility that there's a subgenomic RNA for 3B as well. And we described that in the Periscope manuscript, actually, if you're interested in that. So yeah, so non-canonical subgenomic RNA from sites that don't have a canonical TRS site. And this mutation is a triplet, like I mentioned. So at positions 2, 8, 8, 8, 1, 2, and 3, there are 3 mutations in cis that from a GA, and a G to C that changes this GGG to an AAC. And actually what happens, this actually makes this a TRS site. So the resulting sequence of this mutation is a canonical TRS site. And if you look at isolates with that variant, they have a non-canonical subgenomic RNA in the middle of the nucleocapsid open reading frame. And this has been increasing over time. This was back in March to June last year, where you see the D614G variant in spike was increasing over time. So the blue is D, so it's wild type at that position, and the RG, the triplet mutation I'm talking about here, this is the wild type for that as well. You see it's reducing over time. You see the D614G mutation is in green with this KR triplet, and you can see up to June last year, it was pretty much it was taking over the worldwide sequences. And kind of really started looking at what this was doing to the subgenomic RNA profile. So not only is this mutation producing a new non-canonical subgenomic RNA, actually the total subgenomic RNA in these isolates was also increased. So on the left here, this is all subgenomic RNAs normalized again to genomic, and you can see in the KR, the triplet mutant virus, you can see an increase in the total subgenomic RNA, and it's really restricted to a few open reading frames. So the N subgenomic RNA, M, E, and OF6, but they're very small effects. It's kind of enough to be statistically significant because we've got a lot of sample numbers, but it's not as impressive as some of the changes to subgenomic RNA we see with the newer variants, which I'll talk about in a few slides. So we kind of whet our appetite, right? We'd got involved in this project where we'd seen a variant in SARS-CoV-2 could affect subgenomic RNA by producing a non-canonical subgenomic RNA and changing abundances of subgenomic RNAs. And so we've always kind of wanted to look more widely across the SARS-CoV-2 lineages at what are these variants doing to subgenomic RNA. And really, we really only had enough data to do this after Christmas and after we'd sequenced enough B.1.1.7 viruses. So we've got this preprint out at the moment that describes this work. It's looking at subgenomic RNA specifically in B.1.1.7 compared to all the parental lineages that are out there. So B.1.1.7 became dominant in Sheffield in early 2021. So this again is the same plot I showed you earlier at our sequencing numbers, but split, but colored based on lineage. So you can see in Sheffield, we mostly had B lineage SARS-CoV-2, and I think that's the same for the rest of the UK. And, in the middle of last year, we were seeing dominance of this variant, B.1.1.7.7, not defined as a variant of concern, but had accumulated some interesting mutations. And then towards Christmas and the end of 2020 into 2021, we see the emergence of B.1.1.7. And really, this is all we sequence now in the UK. Pretty much 100% of our sequences are B.1.1.7. And we first started looking at the differences between 1.1.7 and B.1.1.7.7 because that was the previous most frequent lineage that we were sequencing in Sheffield. And we wanted to look at whether we could recapitulate some of the analysis that had been presented in the literature, where people had seen a reduction in ECT. So E gene cycle threshold is the qPCR test measure of how much virus is in that sample. So the lower this is, the more virus or the more viral genomes are present in that sample. So you see a nice statistically significant reduction in the ECT in B.1.1.7 versus the previous lineage. And we see the same for relative light units. So the diagnostic lab here in Sheffield switched over to a different platform. So this is, I think, Hologic Panther, and they measure SARS-CoV-2 genome in relative light units, and you can see an increase in relative light units in the B.1.1.7 lineage. So this was nice. We could see the same effect that people in the literature were reporting, that possibly this relates to a higher viral load in B.1.1.7. The next thing we looked at is subgenomic RNA. So there's two plots on this slide. On the left is pillar 1 and on the right is pillar 2. So pillar 1 is sequencing from our local hospital. So the pipeline I talked about at the start of the presentation, where we're getting samples from our local hospital. So this is patients and healthcare workers. And then pillar 2 is testing from the community. So this is people who have walked into a testing facility in the UK from the community and had a test done, and then we've sequenced their SARS-CoV-2 genome. So I've just picked out the 5 most highly expressed open reading frames from what we know from the previous slides, S, E, M, N, and ORF6. And you can see there's quite an impressive increase in the amount of spike subgenomic RNA in B.1.1.7 infections. Again, normalized to genomic reads. We also see a substantial increase in N, and then to a lesser extent E and M. And we see some of these changes in the pillar 2 data too. So it's not just a function of the different R-specific testing regime in Sheffield, for example. We see that across different pillars of testing. So, we still see a large increase in the amount of spike and N subgenomic RNA in that pillar of testing too. So more subgenomic RNA in B.1.1.7 compared to B.1.1.7.7. And interestingly, if you go back to the parental lineages of SARS-CoV-2, so if we start with this plot on the left, B.1.1.7 is on the right, and all its parental lineages are on the left. So we go B.1.1.7, B.1.1, B.1, and B. And you can see overall the previous parental lineages of B.1.1.7, we see a significant increase in subgenomic RNA for N, M, S, E, and ORF6 as well. And there might be a hint that, some of these lineages are starting to get a little bit more subgenomic RNA than the previous, especially for nucleocapsid, for instance. So we see a statistically significant difference between B1 and B1.1, for example. What's also, I think, really interesting, if you compare B.1.1.7.7 to its parental lineages, especially for ORF M, so membrane and nucleocapsid, those viruses, B.1.1.7.7 viruses, do show an increase in subgenomic RNA over their parental lineages too. So over time, SARS-CoV-2 is obviously gaining mutations that might not make it a variant of concerns, and not the spike mutations that everyone's worried about, but there's definitely something going on with the expression of its genome, even though these spike mutations might not be present in these lineages. So I think that's really interesting. And then a lot of people will say, well, you're looking at RNA from these viruses, but they're more transmissible, they've got higher viral loads, these patients. So is it just a fact that you've got more RNA present? And we can look at, subgenomic RNA versus E-gene cycle threshold, and we see no correlation. So here is the total raw subgenomic RNA, so just the amount of reads we detect in these samples that are subgenomic RNA. And then E-gene cycle threshold, we see no correlation in either lineage. But we do see a correlation with genomic, and that's kind of what you would expect, because these tests are measuring total RNA in these cells, and there's much more genomic RNA than subgenomic RNA. We see a higher correlation, an R of minus 0.4 with a high p-value here, a negative correlation, which you'd expect. So lower ECT means more virus, more genomic RNA present, and we see that from this data. So we think, it shows that we don't really see a correlation with E-gene cycle threshold or the diagnostic test and subgenomic RNA. We see a slight correlation with genomic. So we thought we'd normalize to ECT just to make sure that we weren't seeing an effect of just a higher viral load. And, even doing this, so we multiplied the number of subgenomic RNAs by the E-gene cycle threshold, and we still see a significant increase in the amount of S and N subgenomic RNA. Interestingly, no, we don't see one for E anymore, so maybe this even is cancelling out that effect by normalizing to ECT, which I think is quite interesting. So more subgenomic RNA in B.1.1.7, and it's independent of E gene cycle threshold. And across lineage families, we're seeing an increase in some subgenomic RNAs too. But what if this is just due to a shift in the infection course of these individuals? So there are some papers out there that say that, you know, it's a faster infection, so maybe there's more subgenomic RNA earlier, but in total there's not more subgenomic RNA. So we looked at the amount of time someone has had symptoms before they got tested. So on the day they were tested, how long have you had symptoms? And we had quite a lot of this data. So 398 samples for B.1.1.7 and 334 for B.1.1.7. A lesser amount for some of the other lineages, but I don't really show any analysis of these. But what's interesting, I think, is that you can see from this that there's possibly a shift to the left for B.1.1.7. So, you know, people are presenting a little bit earlier in their infections to get tested, which I think is quite interesting. And we can then treat this like a pseudo-infection time course. So if we plot days of symptom onset on the x-axis and things like relative light units on the y-axis, we can see how that changes over time. And I don't think there's particularly any difference here in the two lineages, but again, we just see higher relative light units in B.1.1.7 infections. But if we look at subgenomic RNA, I think there's quite a striking change. So on the left is B.1.1.7, on the right is B.1.1.7.7. And you can see across the whole time course, really B.1.1.7.7 is kind of flat, with maybe a peak around day 4 or 5, especially with ORF6, the green one here. And if you look at B.1.1.7, there seems to be a shift in that peak of subgenomic RNA abundance to the left of the plot. And you can see, especially for our ORF6, we're seeing this really striking peak at day 1. So it looks like not only is are the variants that, or the SNPs that are present, SNPs, indels, and changes in B.1.1.7 altering the amount of subgenomic RNA, but it's also they're also affecting the kinetics of subgenomic RNA expression in these samples. And we want to make sure that this any change in the days of symptom onset data is not affecting our predictions of our result of the abundance of subgenomic RNA. So we applied a generalized linear model using the reference lineages B1 and showing just this as an effect plot here. So you can see that the most effect on the subgenomic RNA expression of these open reading frames is either being in a 117 lineage or for ORF-M being a 1177 lineage, and actually very little effect from the days of symptom onset. So that's reassuring that even though we see a change in the kinetics, the days of symptom onset is not affecting our conclusion that there is more subgenomic RNA in B.1.1.7 infections. So on to the last part of the talk now. And I think probably one of the most interesting parts is we actually see overrepresentation of a non-canonical subgenomic RNA in B.1.1.7. So remember back to the start of the talk where I talked about this Cta mutant that had a non-canonical subgenomic RNA. A mutation in B.1.1.7 also results in a non-canonical subgenomic RNA. And we didn't go about looking for this actually, so we kind of produced these plots as standard when we're looking at subgenomic RNA. On the top is B.1.1.7.7, on the bottom is B.1.1.7. This is just the SARS-CoV-2 genome, and each one of these arcs is representing a detected non-canonical subgenomic RNA. And the little dot at the end of each arc is the number of samples, which is the color. So the redder this is, the more samples. And the size is the number of reads. And what jumped out to me straight away was at the start of N was this massive red blob that indicates there's something really going on at the start of the N ORF in B.1.1.7 that's not happening in B.1.1.7.7. So it's really exciting finding. And actually, if you look in all the other lineages, so I think there's around 3,800 samples in our other lineage category here, there's a hint of some subgenomic RNA at this position. But if you look at B.1.1.7, there really is an impressive peak of expression of subgenomic RNA at this position, and it's restricted to this lineage. And if you plot it a slightly different way with a box plot, it's highly statistically significant, so that B.1.1.7 really expressed this non-canonical subgenomic RNA to really high levels. But what I want you to remember, though, is that there are some viruses that are not B.1.1.7 that do have some of this non-canonical subgenomic RNA. So then if you look on the right, this is just a zoom in of the plot I showed you on the last slide. The bottom again is B.1.1.7. This CTA here, this red CTA, is actually a triplet mutation that's present in the nucleocapsid open reading frame of SARS-CoV-2. So it's GAT in the normal, in the B.1.1.7 virus. GAT goes to CTA. And this mutation is the D3L mutation in B.1.1.7 lineages. So you can see this CTA and this red dot here. This is where this non-canonical subgenomic RNA is produced from. So just to look at this a bit in more detail, this is a non-B.1.1.7 sample on this slide. So B.1.1.7 has this triplet mutation at D3 in nucleocapsid. It's a D3 to L, and it's a change from this GAT to CTA. So if you look at here at this IGV plot, in this non-B.1.1.7 sample, this is genomic RNA at the top. You see no CTA mutation. We do see some non-canonical subgenomic RNA at this position. Interesting, if you look at the sequence, there's a CTA in this subgenomic RNA, which is interesting, right? So this is the normal. This is the NTRS site here. So you can see it's a little more 5'. This is the nucleocapsid methionine and the ORF9b methionine. You can see that this non-canonical subgenomic RNA misses the nucleocapsid methionine, but would include this ORF9b methionine. So if you look at a B.1.1.7 sample, on the top again, I'm plotting non-canonical subgenomic RNA. At the bottom, I've got the canonical subgenomic RNA for N. So this is what we normally see for N. This is the leader sequence, all these bases here. This is the NTRS site and the methionine. And because this is a B.1.1.7 sample, we have the D3L mutation, right? So a CTA at this position. And then you see more non-canonical subgenomic RNA in these B.1.1.7 samples. And again, this has got this CTA mutation. Apologies, I've just clicked the wrong button. It's got this CTA mutation. So what we think's happening, and why there is more of this non-canonical subgenomic RNA in B.1.1.7 samples, is this CTA mutation has actually increased the complementarity of that region to the leader sequence, making RDRP produce a subgenomic RNA from this position more often. So in this plot here, the genomic leader is the top sequence here. This is the TRS-L site that I talked about right at the start of this talk. The bottom one is the genomic sequence. So here's the CTA, CUA mutation if we're speaking in RNA. This is the nucleocapsid D3L. In the middle is our nascent subgenomic RNA template, so our negative stranded subgenomic RNA. So RDRP jumps more often from this position because this CTA is present at this position in the genomic leader, and we have more complementarity. So the normal complementarity is in this region is the gray lines, and the red lines are a result of this mutation. So you can see that this CTA matches the genomic leader more than wild type would, or non-B.1.1.7 SARS-CoV-2 virus. And so in the normal situation, so a non-B.1.1.7 sample, our hypothesis is that you get a low level, and we see this in the data, right? I showed you that non-B.1.1.7 lineages, there was a very, very low level of this non-canonical subgenomic RNA. So what we think happens normally is that you've got this low amount of complementarity in this region. So you get the odd read or the odd non-canonical subgenomic RNA produced that has this CTA mutation in, this CTA, sorry, not a mutation in this case, but it has CTA that is a contribution from the genomic leader sequence. So here we go, this is the plot again. We get RDRP, it's synthesizing this strand from the genomic RNA, it jumps over to the leader and produces this non-canonical subgenomic RNA that has in the template GAU, and then when that gets converted into a positive strand, it has this CTA. So that's what we saw from the reads, right? We could see that CTA mutation in this non-canonical subgenomic RNA. So that's the normal sample. And what we think happened, actually, this CTA mutation was actually produced from by recombination between this non-canonical subgenomic RNA for ORF9b and the genome. So this has been discussed in other viruses where you have this recombination method called copy choice. So in this case, RNA-dependent RNA polymerase starts transcribing a negative stranded RNA using our non-canonical ORF9b subgenomic RNA as a template. It then gets this CTA, it includes that in the template, and then switches strands to the genome and finishes off its negative stranded RNA on the genome. So then you've got this molecule, this RNA molecule, that is a result of both ORF9b non-canonical subgenomic RNA with this CTA at this position and the genome. So you get the negative strand genomic RNA produced and then positive strand synthesis, which results in this CTA being fixed in this genome. And for some reason, this virus is outcompeted and the CTA mutations become fixed in this lineage. So we think this is really pretty strong evidence that a non-canonical subgenomic RNA has been a template for RDRP and a recombination that has resulted in this CTA mutation, which subsequently, results in the production of more ORF9b subgenomic RNA. Or in a subgenomic RNA that could represent ORF9b. We have no evidence that there is ORF9b produced from this subgenomic RNA, and that's work that needs to be done, I think, in the future. But it's interesting because ORF9b has been shown in the literature to suppress the interferon response. So it really starts you thinking whether B.1.1.7 is really producing more proteins that can affect or modulate immune response early in infection to allow it to produce more virus and be more transmissible, more severe, etc., that we've seen of this lineage. So it'd be really nice to confirm that we get more ORF9b protein produced as a result of this mutation, and then see how this affects transmissibility, etc. So that's it. I hope you've enjoyed the journey through subgenomic RNA as much as I have learning about subgenomic RNA, because I'd never heard of subgenomic RNA before last spring. But I hope I've shown you that we can detect subgenomic RNA from genomic sequencing data, and that B.1.1.7 virus has more subgenomic RNA than previous lineages. And it has those subgenomic RNAs earlier in infection than the previous lineages. And I think that one of the most interesting findings is this enrichment of this subgenomic RNA from that could represent ORF9b as a direct result of this triplet mutation. And I showed you data of other triplet mutations. So it starts to become interesting that are there other triplet mutations in SARS-CoV-2 being formed in this way through recombination between subgenomic RNA and the genome? And I think it's important that we should focus on these mutations, that not only spike mutations are important in SARS-CoV-2 biology, but also mutations in other genes that can obviously affect expression of the genome and therefore the phenotype of these viruses. And actually then it becomes quite interesting. You think about maybe we could even start predicting new variants, right? We know all the non-canonical subgenomic RNAs being produced. So could we look at these sites and determine likely triplet mutations that could occur, or sites that might be more susceptible to mutation because they're producing a lot of non-canonical subgenomic RNA. So I think that's interesting work for the future. So thanks all for listening. Just one more slide, and we're recruiting actually, and we're recruiting a bioinformatics developer to help us further develop Periscope and integrate it into our national surveillance pipelines, and try and make a web application out of Periscope so that people can just easily run this on their data. So yeah, informal inquiries are welcome. So send me a tweet or drop me an email, and and we can talk about what the position involves. So thank you all for listening, and thanks to all the collaborators, all the people involved in Sheffield and in COG-UK. Thank you.
Thanks, Matt. That was a great talk, and indeed we enjoyed this journey into hearing about subgenomic RNA. Of course, a great talk always brings in great questions. So question time. Audience, a gentle reminder that if you have questions for Matt, please type them in the Q&A box. We will call you to ask your question, but we're also happy to read out your question. So as you're waiting for the audience, let me get started with the question. So do you know what the lifespan of the subgenomic RNA is? I mean, are they by any means indicative of an active infection, or are they stable enough to hang around for a while?
So there are a few papers on this, and I think, on one hand, I think they do hang around. I think people have shown that they are bound to some membranes in the cell, so they can hang around for quite a while. But we did do that infection time course, and I know this is in vitro data and not in vivo, but we do see a drop in subgenomic RNA too. So we see increases and we see decreases in the amount of subgenomic RNA present. So there obviously is an accumulation, and people have shown that in the literature. But I think, like any RNA, it is also degraded and chewed up. So there is some flux in the amount of subgenomic RNA present, I guess. And so, yeah, I think the jury's still out on whether it represents an active infection. I think what we've seen I didn't show the data here, but we've had patients who have been positive for a very, very long time, and we've took samples throughout their infection, or at least their, in some cases probably a false positive. It is RNA hanging around. But what you see is a real reduction in the diversity of subgenomic RNA in those samples. So we still see some subgenomic RNA present, but we don't see all of them. So they might have the full complement at the start of their infection, but in samples that are 6 weeks, 12 weeks down the line, we still see some subgenomic RNA, but we see a very much reduced diversity in the subgenomic RNAs that we can detect.
Right. Thanks.
Matt, amazing talk.
Thank you.
Got a lot of questions. I'm going to start with something just really remedial that I'm not certain about. So if RDRP is transcribing from the 3' end, to make the longer subgenomic RNAs, does it end up like reading through some of those TRSs?
Yes. Yeah. So you will. It doesn't always jump, and I don't think we know why it jumps at some rather than others. And you might even expect that you'd see more jumping earlier on. And I think some of the direct RNA-seq experiments, you see higher coverage at the 3' end of the genome, indicating you're more likely to see. But then we see very high expression of spike, right, which is the longest subgenomic RNA.
Yeah, so that's what's driving me crazy. Is why aren't the subgenomic RNAs greatly biased towards the 3' end? I guess, I get it, that was sequencing, but a lot of sequencing itself is 3' biased.
And also you're going to bias towards the shorter fragments. So I don't think that question, I don't know if there's an answer for that, at least that I know of. There might be, but I don't know. And it's sometimes, the TRS sites for most of the RFs are the canonical sites. Some have a slight reduction, and they're not the canonical. But I think I don't know. I think the jury's still out on why that happens.
Yeah. And wait, to follow up. So you said some people have just done less biased RNA-seq, trying to figure out the full complement of subgenomic RNAs. So how does that match up with, so you said your sequencing is amplifying things with the leader, but RNA-seq would find leaderless subgenomic? Yeah.
So there is a paper that the architecture of the SARS-CoV-2 transcriptome, a really nice Cell paper. Can't remember the first author now, but they bin these reads into different bins, and you obviously, RDRP is jumping all over the place all the time. I think that's one of the features, and that enables recombination and mutation formation. So you have leaderless subgenomic RNAs, or I don't know if they're actually subgenomic RNA. Well, they are shorter than the genome, so I guess they are. But I think the way we think about it is these are the kind of classical subgenomic RNAs with leader, and we really can't detect the other events. We probably would see those in the genomic data, right? We would see them as kind of structural variants or big regions of the genome missing, but they're much more difficult to detect, and it's much more difficult to separate those out from artifacts introduced by chimera formation and stuff when you do library prep. Sorry, but I cut you off to the punchline of your question. Was that the answer you were looking for before I jumped in?
I think so.
Okay. Yeah.
So Matt, I did some quick Googling while you were talking, trying to figure out, like, how can we figure out the functional consequences of all of these subgenomic RNAs? And by that, I'm sure you saw this paper out by an Israeli group end of last year that did ribosome profiling. And so, there you're finding RNAs that have been recruited to the ribosome. You don't know they're getting translated, but at least they've made it through the door. And so they found some non-canonical subgenomic RNAs. Did those match up well with yours? And is there like a bias of which ones are docking to the ribosome and which ones may not be?
I've not done that overlap. It's been on my list of things to do. I downloaded some of that supplementary material to look at that, but I've not got around to that yet. But you're right. I think so. We see these reproducibly, right? And in our data, the Glasgow data, the cell line data, we see these non-canonical subgenomic RNAs from these sites that are reproducible and a bit more stochastic as well. So I think the jury's out on what they do. I think some people think they just, over time, you just accumulate all this stuff that RDRP is doing and not really doing anything. And I guess that's the question they're answering by doing the ribosome docked, which of these actually are getting translated. So I need to do that overlap analysis and see which of ours overlap. But yeah, I think they found similar things, right? They didn't see much ORF10 in there, which I think most people agree on that whatever method they've used, no one's really seen much ORF10, not much ORF8. So yeah, I think everyone's kind of coming to a conclusion that these two are not expressed that highly, at least.
Yeah. And do you know if anyone's done mass spec to see like the last step of which things actually are translated and persistent?
I think someone did, and I've cited it. I can't remember what paper it is at the moment. I don't know if it's I'll have to have a look at my notes, but there is some proteome analysis, I believe, and it's either a Science or a Nature paper, pretty big paper of profiling it. I know that they're broadly, I looked at that because they agreed with what we were seeing, on the subgenomic RNA front. So I think there is some data out there on that.
Mm-hmm. Okay, Sarada has one again, and audiences. So Sarada, you go, and then we'll call on Limor.
Limor, yeah. So Matt, my question is: do you see any correlation between the abundance of both subgenomic and non-canonical subgenomic RNA and the intensity of infection?
So what do you mean by intensity? So some kind of clinical,
asymptomatic or severe infections? Or is it even possible to look at that granular level given the high throughput?
Yeah, it's something always been at the back of our minds, but it's very difficult to collect enough of that data to make a conclusion, right? I think, as I said at the start of the talk, I think, our method is a byproduct of the genomic sequencing, right? So do I think it's perfect method for subgenomic RNA measurement? I don't. So I think our strength is numbers. So, with the B.1.1.7 work, we have so many data points that we can see this effect of B.1.1.7 in our subgenomic RNA data from the genomic sequencing. So we need numbers, and we need numbers of both that and the clinical data, which is difficult to come by. So I think it's something we want to do, but we're not there yet on numbers. And actually, I think for the positive thing for all of us is that the numbers are dropping, and so hopefully we never got enough data to do that analysis, right?
Well, that's a good problem to have.
Yeah, I think it is a good problem to have.
All right, thank you. And Limor, could you please unmute yourself and ask your question?
That was really nice talk. I was wondering if there's any importance to the ratio of the different subgenomic RNAs. I noticed that the N and S seem to go up more than the others.
Yeah.
Does that,
And that's something I think people think about non-canonical subgenomic RNA. If it doesn't have a function, maybe it tips the balance of transcription away from So I think there is literature out there that discusses this balance in subgenomic RNA production, and it obviously has an effect on what's available to produce virons. So I don't know, but I've got a feeling the ratios probably do matter, but we don't have any data to support that or really even offer a hypothesis for that. But you're right. I think there probably is something in that. And, if it's, B.1.1.7, for instance, we didn't really look at it before, but as I was preparing this talk and practicing, I did keep seeing this M, and a really striking increase in M in that lineage. So it's something I want to go back to and find out why that might be. But yeah, I think there probably is something in the ratio of different subgenomic RNAs, and maybe that's a measure we should start producing from Periscope is the ratios. And actually, then that gets around the problem of, abundance of specific ORFs, like I talked about RF7A. Maybe if we do ratios, we don't need to know the final level, and actually that ratio might be more informative. It's something, that's a really interesting point. We should do that. Yeah.
Thanks.
Thank you.
Thanks, Timur.
Matt, random thought. So people aren't seeing much, but very high levels of ORF8 and ORF10. But they're pretty conserved and they're not really being eliminated. Do you think they have a non-coding function?
I don't know, but the also important thing, I think I always forget this, and the important thing I think to remember is that all the other subgenomic RNAs will have ORF10 in there, right, and 8. So just because they, I kind of forget this, but because they don't have their own subgenomic RNA doesn't mean they're not producing protein, right? It could be that enough subgenomic RNA for enough protein products is produced from the other open reading frames that they don't need their own.
Yeah, you're right.
Or the ratio, the ratio is tipped about in a balance that it's not required. So it's something I always forget actually that the subgenomic RNA from S, it actually includes all the downstream ones. And I spent absolutely ages thinking about this and thinking about a better normalization method, because if you think about it, when you're doing genomic sequencing, all those downstream amplicons could be amplifying from subgenomic RNA, because that S subgenomic RNA includes all the downstream genomic material. So we started getting into this, and I'm glad I abandoned it, because we got into this complicated normalization procedure of kind of like cumulative expression of these. So the S open reading frame, the N, they could include, so it got complicated. I spent ages. I made a really nice figure, and then I just abandoned it because we decided it didn't really add that much. But yeah, so they could be producing protein from other open, and the genomic RNA too. So just because they don't have their own subgenomic, they might be expressing protein.
Yeah.
And that's what people see from other SARS, right? That ORF8 got this big deletion and it became less virulent, and that's what people think might be the reason that happened. At least that's what, as I recall, that's what people think. So, they probably do have an important function. It's just where they don't have their own subgenomic RNA, at least that we can detect from the methods that everyone's been using.
Yeah.
Yeah.
Yeah. Wow. Gosh, okay, this is a lot to think about. Yeah,
it's been a crazy ride, right? I think we really got involved through that group, and then we developed this tool just to do the analysis for that paper, really, and then we discovered that we could really, actually it's a lot of powerful data that was otherwise being thrown away, and now we want to try and integrate this into the national framework. So if variants do come along, we can see these changes immediately, right? We can see an increase in subgenomic RNA. And maybe it's just going to be incremental like we see across the lineages, but maybe there'll be a big jump, and maybe we'll spot something. It's an instant phenotypic readout that we can get from data that's already being produced. So we might as well do it, right? Even if we don't see anything, even if we don't have the numbers to see a new variant of concern and some, change to subgenomic RNA. But we'll have a look, and I think it's important to get this phenotypic readout as early as we can. So yeah, it's been interesting for sure.
Yeah, I'll just say that, even though you don't know exactly what's going on, that that incremental increase of the subgenomic RNA levels from the B.1.1.7 lineage, that's pretty creepy, clearly something is happening, and we may not be seeing the effects from it yet.
Yeah, And even, you know, even if it is just a proxy for a change in the infection time course, and people are more infectious early, they have more virus earlier, that's cool that we can see that from the subgenomic RNA. I think, you know, it adds more information from just e-gene cycle threshold and other, you know, diagnostic tests. We can use this subgenomic now, be a bit more granular and see these changes, even if it's not a change in subgenomic RNA expression, abundance, kinetics, whatever. You know, it's an additional measure than the diagnostic test. So I think it's interesting, even if those things don't pan out, you know. It's just another metric that we can measure that gives us more information about this virus, and, you know, information's power, I guess. So
yeah.
Oh, hey Matt.
Hello.
So speaking of variants of concern, other variants, so you showed us data with respect to B.1.1.7, and agreed that was the dominant variant in the UK. But any chance did you have enough samples from the other variants, the 351 or the P1 variant, to see what's going on?
So we would love to do this, right? And I think the paper, you know, the killer paper is all the Vox together, right? And so if we'd had that data locally, we would have done that. So we're trying to collaborate with people. The problem is, I think that, you know, we're sequencing our runs for 24 hours, so we get a reasonable amount of reads, and we show in the Periscope paper really below 50,000 reads, you start to see really not very good performance of this kind of analysis, because you don't get the diversity, you don't get enough subgenomic RNA reads. I should have really said that you're talking around 1% of the total that is subgenomic. So we've been looking at other people's data sets, and rightly, they're just doing it for genomic surveillance, and they don't have enough reads in some of these data sets. So we are looking at other data sets, and the initial impression is not an increase in subgenomic RNA in those lineages, but I don't really have enough data to say for sure. I mean, deep down, if you held a gun to my head and said, what do you think? I think they don't have D3L, and I think D3L is a really key mutation that's not really getting much attention at the moment. You know, they all share these spike mutations, which are obviously very important. I'm not saying they're not important at all, but there's something with this D3L that is enabling the B.1.1.7 to be, you know, I think would probably in a cage match, I think B.1.1.7 would win those other variants, right? I think they would be outcompeted if it was a fair fight, I think. So very preliminary data, and we hope to have more, so we'll see. But like we saw with B.1.1.7 at the very start, we only had a few samples, and we didn't see too much of a convincing change that definitely with sample numbers really became very apparent. So I think I'll say that, but things could definitely change when we get more data.
Sure.
Yeah.
Thanks.
Yeah, Matt. Okay, that was amazing.
Great. Thank you for having me. It was great to share the work.
Yeah, that was really great. So everyone, join us same time next week. We're still finalizing the exact speaker, but it'll be excellent. So everyone stay safe. All right, see you next week. Bye-bye.